And what I'm going to ask everybody to do, what we always do with these, is just summarize kind of where you think the space is going. I'll start with Carrie.
So I think one of the themes at ASCO this year was, again, de-escalation. I think we're learning more and more about which patients need which therapy. And I think we did learn a lot about what we don't need to do moving forward. I think the Caitlin data told us that trastuzumab, pertuzumab in the adjuvant setting is here to stay. And I think the metastatic space has really been fantastic. I think, leaving San Antonio this year, I think I was the most enthusiastic about a space that I've been in a really long time, thinking about all the new agents that we have for metastatic HER2 positive breast cancer. It makes it more challenging, but that is certainly, I know, a challenge we will all take on because it's going to improve our patients' outcomes. So I think the challenge now is going to be really in the metastatic setting, determining the sequence, determining the right patient for which treatment, and really try to do this in the most logical manner, so that we are able to keep patients stable and living well with their breast cancer for as long as they possibly can in the HER2 positive space.
Good. Mark?
Well, I think, I'm excited by the fact that it's clear now that HER2 ADCs are here to stay. We definitely need more than one. We need more than two, because what we'd like to have is a tool chest of ADCs with non-cross-resistant payloads so that we could use them in sequence, or with different epitopes for the targeting antibody backbone. You could even use them in combination as long as there's not too much overlapping toxicity. The other thing that excites me is the tucatinib data from our colleague here, Dr. Murthy. I mean, it's just spectacular data. So nice to finally have a pure HER2 antagonist. That was the holy grail all the way back to the early lapatinib days, recognizing that the other HER2 TKIs that are approved have off-target toxicities due to EGFR kinase inhibition. So there's huge opportunities there, especially in early stage disease, particularly in the extended adjuvant or post-neoadjuvant setting, indeed, a la Extenet. I think there's a huge opportunity there that I wouldn't want to see lost. And finally, I'm very keen on further antibody engineering, in particular bispecifics. There was a very nice HER2, HER3 bispecific antibody that was presented at this ASCO that looks very, very interesting. It binds to subdomain 1 on the HER2 extracellular domain, so it doesn't bind to the pertuzumab or the trastuzumab epitope, a whole new epitope. And on the HER3 molecule, it binds to HER3 in its tethered conformation, so it can't bind to ligand. So you can't get HER2, HER3 ligand activated activity at all with that molecule. So it's very, very interesting. Zymeworks has a HER2 bispecific that's very potent as a naked antibody in heavily pretreated patients in the phase 1 dose escalation. Very nice and high frequency anecdotal responders. So I think that's really cool. And finally, antibody fusions, where you can fuse other therapeutic moieties to the FC domain of a HER2 antibody and kind of give it whatever attribute you want. An IO attribute, for instance, we're doing that now at Stanford with an investigator-initiated trial from a company called Bolt Therapeutics. It's a new startup. And so that's going to be really interesting to look at the fusions in the future as well.
Virginia?
It was interesting at ASCO, there were two discussions. One is when to escalate, and the other one was when to de-escalate treatment. So obviously we're seeing this more and more in the neoadjuvant, adjuvant setting, where we don't want to overtreat patients. We don't want to undertreat patients either. I think what's interesting for me in the future is the HER2 low. We have so far an ADC that seems to be active in that space. I can't wait to get more results. HER2 mutations, again, we have a DKI that seems to be doing pretty well. I'm sure others will do pretty well as well. So HER2 becomes more than just the HER2 positive disease. It becomes a lot more inclusive, and that's what's exciting me.
Good. And finally, Rashmi?
Yeah, so I echo everyone else's thoughts, but also I would say, I think the most exciting parts for me since San Antonio has been really the approval of all these agents and really the expansion of our toolbox for patients patients in the clinic, which ultimately is going to lead to improved outcomes. I think it's been very exciting to be able to think about systemic treatment options now for patients with brain metastases. And I hope that, you know, the future would allow for us to even think about prevention early on. So to prevent them from having CNS relapse, which really oftentimes drives their overall prognosis in the setting of metastatic disease. And so, you know, I'm looking forward to more studies and more work to kind of further delve into that.
Great. Well, again, thank all of you. This, I think, was really great. And thanks to our viewing audience for listening to this. And we hope you found this OncLive peer exchange discussion to be useful and informative. So again, thank you very much.